Welcome back to the Dairy Knowledge Exchange. I'm Pat Colley, and I'm excited to share a fresh initiative we're launching in 2025 here at D&D Ingredients. Each month, we'll bring an educational webinar on this platform, spotlighting timely industry topics and featuring one of our valued partner products. Make sure to visit the D&D website regularly to explore these new learning opportunities.
Hello, my name is Erin Horst, and I'm a dairy technical consultant with Elanco Animal Health. And in today's webinar, we're going to be talking about this idea of immunosuppression and whether or not it's truly a fair characterization of how functional the immune system is around the time of calving, and whether or not it really contributes to poor transition cow success. It's likely that for many of us tuning into this webinar that we're intimately familiar with the transition period defined as the 3 weeks prior to the 3 weeks post-calving. And we've probably all seen figures like the one depicted here on the right that shows the various changes that are occurring in this relatively short window of time. We see drastic reductions in energy balance, decreases in calcium status, and we've probably all focused a lot on this described reduction in immune cell competence, which has been a huge focus area for understanding what creates poor transition cow success. With all of these changes going on at the same time, it's not necessarily surprising that there is this disproportionate incidence of health problems and herd culling that occurs. And as a result of this, there's been a huge focus in the academic community as well as in industry and really understanding what the underlying mechanisms are that ultimately regulate transition cow success, as this has profound implications to welfare as well as economic profitability. Historically, when we've thought about transition cow success, many of us recognize that there's been a very large focus on how when the degree of negative energy balance is exacerbated, as shown by increases in NEPAs and ketones, as well as when there's a significant reduction in blood calcium status, that these are thought to actually cause poor transition cow performance through their supposed impact on depressing dry matter intake, impairing reproductive performance, and also directly causing a reduction in immune cell functional activity, leading to exacerbated immunosuppression and thus increased risk of infectious disease. I want to start this webinar out today by going through a little bit of a refresher on how this concept of periparturient immunosuppression was first described and how changes in NEPAs, ketones, and calcium were ultimately connected to this concept of immunosuppression. In large part, the experimental designs that were utilized to describe immunosuppression all utilized a similar approach. Essentially, researchers would go out to farms prior to calving in the early postpartum period and then a little bit further into lactation and collect blood samples. Those blood samples were then taken back to the lab and they would isolate specific immune cell populations like neutrophils and macrophages. And they would stimulate those immune cells in vitro and then assess how capable the immune cells were of clearing a pathogen. So their ability to phagocytose, to release reactive oxygen species, and also to migrate in response to a signal like cytokines or chemokines. And essentially what these researchers described is that various aspects of the immune system or of these functional activities were shown to be depressed postpartum relative to prepartum or further into lactation. And so at the same time as this research was going on, researchers were also starting to connect changes in NEPAs and ketones and low levels of blood calcium with poor transition cow success. So they decided to take these studies a step further and start to incubate these immune cells in high levels of NEPAs and ketones. And some researchers found that certain aspects of the immune system were further suppressed in the face of these high levels of NEPAs and ketones. They would then also go out to farms and find the cows that were hyperketonemic or hypocalcemic, collect blood and compare them to otherwise what appeared to be healthy cows. And similar to the in vitro assessments, they said, okay, when these immune cells are stimulated in a petri dish, again, these functional activities appear to be more suppressed in the hyperketonemic and hypocalcemic cows. And so the dogma became that transition cows are immunosuppressed, and that this immunosuppression is exacerbated by greater degrees of negative energy balance and hypocalcemia. So NEPAs, ketones, and calcium actually cause immunosuppression, and that leads to the poor transition cow health. But over the last 15 to 20 years, we've really started to refine our understanding of the immune system. And we've started to question whether or not this idea of immunosuppression is really a fair characterization. And I think one of the really pivotal studies that was first looking into this in a little more detail was a study published by Sabina Mann in 2019, who's now at Cornell. In this study, she utilized a similar approach where she went out to dairies and collected blood samples pre and postpartum, but then to look at functional activity, they actually ran transcriptome analyses to get a more holistic view of the immune system. And the figure on the right actually shows some of the results from this work. And what I want you to focus on are the orange bars relative to the blue bars. So orange bars represent an upregulation in that activity or that functional aspect of the immune system postpartum relative to prepartum. So orange means more activated or more responsive, whereas blue represents a downregulation of that functional activity postpartum relative to prepartum. And what you can quickly see is there are a lot more orange bars. So a lot more aspects of the immune system are actually further activated in the postpartum cow compared to the prepartum cow, which really starts to beg the question or ask us to understand whether or not immunosuppression is really a fair characterization or if we've oversimplified the immune system, because there are a lot of different ways that the immune system functions. One of the reasons we might have found ourselves in this place of oversimplifying in the immune system is the techniques that we have available to really assess immune cell functional activity at the time. Again, we relied a lot on isolating these immune cells and then incubating them with a pathogen or with some sort of immune stimulate and assessing that function. But we miss a larger part of the story when we look in a petri dish as opposed to the whole animal. And I want to highlight a few of the really important areas by which these in vitro assays may be limiting our bigger picture of the immune system. And the first of these that I want to highlight is this idea of immune cell recruitment or movement from circulation into tissue. We know that in response to an immune challenge, there's this need for immune cells to migrate to sites of infection. And we see that this can be observed in response to models of immune activation, such as utilizing systemic LPS or intravenous LPS infusions. When we induce an LPS challenge, we see that there is this very stereotypical response in circulating immune cells, where initially the cell counts within circulation significantly decline and then eventually increase within the bloodstream. And this initial decrease in cell counts is likely explained by migration of those immune cells into tissue, whereas this increase phase is actually likely to be an increase in release of immune cells from the bone marrow. So the bone marrow is trying to replenish that immune cell population. Changes in immune cell counts within circulation can also be observed around the time of calving, where we see this large peak just prior or around the time of calving, and then a significant decline as those immune cells are getting into tissue. And this dynamic change in immune cell counts is really important because the functional activity of immune cells is changing as this dynamic change is occurring. And this is especially true during inflammatory events like the transition period. During inflammatory events, when the bone marrow needs to restore the immune cell population, it will release a greater proportion of immune cells that are immature or less competent. Now, this may sound to you like a suppressed immune system, right? Those are less mature immune cells because the bone marrow needed to release them. But the reality is that it's likely that the immune cells that are in tissue, those immune cells that first migrated out, that signaled to the bone marrow that it was time to release some more immune cells, those immune cells are highly functional, out doing their job. And the immune cells that are in circulation in that given time are still maturing. They will continue to mature while they are in the circulation. And then when they're needed at the tissue, they'll likely be in that mature state and can be functional. To kind of better illustrate this concept, I pulled a figure that was created by Dr. Julie Oppengorf and Dr. Lance Baumgart during her PhD work with Lance. And I think it does a really good job of depicting why this is such an important concept. When we look at cows prepartum, as I talked about before, the idea was that the immune system within prepartum cows remained highly functional. So we know that prior to calving, there are probably less immune insults occurring in that animal. And so there's probably not a lot of migration of these immune cells into tissue. So when we go to take a blood sample from a cow prepartum, it's likely that the majority of immune cells that we're going to isolate from the bloodstream are highly functional, mature immune cells. Now, after calving, the story changes. Right after calving, there's going to be a large recruitment of immune cells into the sites of infection, particularly the mammary gland, uterus, and probably the gut. And because there's this migration of immune cells into tissue, the bone marrow signal that it needs to release more immune cells. And again, it's likely because that animal is in an inflammatory state that a lot of these immune cells are actually going to be immature. Now, this does not say that the immune system at that given time isn't functional. The immune cells that were ready to do their job are off doing their job. The immune cells that we're looking at in the bloodstream are our second line of defense. They're still developing. They're going to gain that skill set over time. But luckily, our first soldiers are already out there fighting the battle. So it's not that that cow doesn't have a functional immune system. It's just the cells that are in circulation aren't representative of the immune system that's actually doing the job at the given moment. So when we collect that blood sample and it's a majority of immature cells, overall, the immune system looks less functional through these techniques. And so this is a really important concept when thinking through how we take in vitro data and apply it to an in vivo animal. Another really important thing to consider when extrapolating our in vitro findings to an in vivo animal is the metabolism of the immune system. And this is an area that, again, has developed quite a bit within the dairy science community over the last 10 years. We now have a good appreciation for the fact that when immune cells become activated, they become obligated glucose utilizers, and they start to ramp up how much glucose they use by a significant degree. And with that increase in glucose utilization by the immune system, we also see that there are dynamic shifts in various aspects of nutrient partitioning or energy partitioning that occur in order to ensure that glucose can be made available for the immune system. So we see a significant increase in insulin, mobilization of tissue reserves, etc. And so this is a really important concept because we see that there are these very intricate changes in energy metabolism that are very likely difficult to apply in a petri dish, right? So we may actually be causing immune cells in vitro to utilize fuel sources that they normally wouldn't because we're not supplying that same metabolic or hormonal environment in that setting. And so that could ultimately go on to influence how functional those immune cells appear to be. Because of these limitations, as well as others that I don't have time to get into, we have to be very careful utilizing in vitro techniques to really get an assessment of the overall immune system. But we need to assess how immune cell function differs in an early lactation cow compared to a mid-lactation cow so that we can better refine how we help the immune system or how we help a transition cow be more successful, especially if immunosuppression is one of the reasons why these cows are struggling. And so recently, there was a set of papers published out of Lance Baumgart's lab that really sought to answer this question in an in vivo model. So in this study, the objective was to evaluate the immune and inflammatory responses to LPS in early compared to mid-lactation cows. And the hypothesis was that early lactation cows would have a diminished immune and inflammatory response to LPS, for one, because we've presumed that the immune system is suppressed in early lactation for quite some time now. And two, because we think it's likely that a cow in early lactation is more tolerant to LPS because she's likely experienced more LPS exposure at that given time compared to a mid-lactation cow. These are the two studies that are a part one and a part two of the first study that we'll talk through, which are both published in the Journal of Dairy Science. Again, this was Julie Oppengorst's PhD work when she was at Iowa State with Lance Baumgart. Great studies if you want to check these out, but we're going to go through a few of the key findings from these results or from this work. In the studies, there were 23 cows that were enrolled in a 2x2 factorial design. So first factor was early or mid-lactation, so lactation stage, and then they either received LPS or they were pair-fed. The pair-fed was just to eliminate differences in dry matter intake, and they received saline. For the sake of time in this presentation, we're really only going to be focusing in on the response to LPS, which is going to be shown in the blue and the tan triangles. So on these figures, the y-axis is the variable of interest, the x-axis is the time relative to LPS. The blue triangles with the dashed lines are LPS cows in early lactation, and then the tan dashed triangle lines are those that were in mid-lactation and received an LPS challenge. And so the first variable we're looking at is the febrile response to LPS. As we would expect after we give LPS, there's a significant increase in rectal temperature, and that was observed in both treatments, both the early and mid-lactation cow. But the degree of increase in rectal temperature was far greater in the early lactation cow compared to the mid-lactation cow. So this is really interesting because we know oftentimes rectal temperature is a downstream response of things like inflammatory cytokines released by immune cells. So this increased febrile response is really interesting. And the increase in febrile response actually is very fitting with what was observed in inflammatory cytokines. So in this study, they looked at both pro and anti-inflammatory cytokines. And the pro-inflammatory cytokines, TNF-alpha and IL-6, were actually shown to be increased to a greater degree in early lactation cows compared to mid-lactation. Again, cytokines are often released by immune cells as well as other epithelial cells, and they act as the cell signal to amplify inflammation, to alert the body that, hey, there's a challenge going on, we need to mount an immune response. And they're also involved in signaling migration of immune cells. So it's interesting to see that cows in early lactation are mounting a greater cytokine response. And this isn't due to pre-existing differences. This is truly unique to that individual challenge. Interestingly, just like the pro-inflammatory cytokines, the anti-inflammatory cytokine IL-10 was also shown to be upregulated in early lactation compared to the mid-lactation cow following LPS. Consistent with the increase in pro-inflammatory cytokines, they also saw that the inflammatory acute phase proteins, LPS binding protein, haptoglobin, and serum amyloid A, were all increased to a greater extent in the early lactation cow compared to the mid-lactation cow. Again, this isn't explained by the fact that they were more inflamed before the LPS challenge. This is truly unique to that LPS challenge. These acute phase proteins are really our gold standard for assessing inflammatory state in a transition cow. So we're seeing, again, more robust immune responsiveness, more robust inflammation in early compared to mid-lactation cows. In addition to this, those metrics, the researchers also evaluated circulating immune cell counts, neutrophils, and monocytes. They saw the stereotypical pattern where initially cell number declined, again, likely due to migration into tissue. And then eventually the increase was observed in both treatments, but the increase was far greater in early lactation cows compared to mid-lactation cows. So these early lactation cows appear to be more primed for releasing those immune cells from the bone marrow when compared to the mid-lactation cow. So this is really interesting to see that, again, just greater release of immune cells. The last variable I'll go in today in this webinar is ionized calcium concentrations. So we've learned through time that there's a very tight relationship between immune activation and calcium. When we induce immune activation, calcium levels plummet. It's very consistent across species. And that was again observed in this study. But what was interesting is that the decrease in ionized calcium was greater in the early lactation cow compared to mid-lactation. Again, it's really important to note this isn't explained by the onset of milk production, right? These cows were already 21 days in milk, but this is likely a reflection of a greater immune response or a more robust immune response in early lactation. So the summary of these results shows that LPS again induced our stereotypical responses. It's a very repeatable model. It's not perfect for assessing immune activation because it's a potent challenge, but we see very, very stereotypical responses, which gives us a lot of faith in the model. But in complete opposite, in complete contrast to the hypothesis, what these researchers showed is that there was actually a greater immune response in early lactation cows compared to mid-lactation, which really challenges this idea that early lactation cows are immunosuppressed. They are very capable of mounting an immune response to an LPS challenge, and in fact, it's a greater response when compared to mid-lactation. Lance's group also then went on to ask the same question using an intramammary LPS challenge model. We know that intravenous is not as realistic, right? So this was meant to be a more realistic model of an immune event that could occur. And in short summary, what they saw is very similar responses to the intravenous challenge. Cows in early lactation had a more severe febrile response, greater increases in cytokines, didn't show the chemotoxic cytokines, but they were also more upregulated, increases in acute phase proteins, more drastic decline in ionized calcium, as well as changes in energy metabolism and production variables that we don't have time to get into today. So just a second example that these cows are, this is consistent across studies and across modes of administration for the immune challenge, that these early lactation cows are mounting a more robust response. I did want to pull out a few graphs from this study with the intramammary challenge. Just like in the intravenous challenge, we see that the neutrophil counts were increased to a greater extent in that secondary phase in early lactation cows compared to mid-lactation cows. But they were actually equally as capable of getting into tissue. So this increase in cell count wasn't explained by a failure of those immune cells to get into sites of infection. And this is a really important point because early on, when developing this concept of immunosuppression, it was really thought that the failure of immune cells to get into tissue was one of the reasons why infectious disease was higher in the transition period, and that connection between immunosuppression and transition cow health. So these results provide really strong evidence that we need to rethink this idea of immunosuppression. But it also begs the question, if cows aren't immunosuppressed, why exactly is there this increased risk of disease? And we think the answer to that question is actually pretty simple. And it comes back to the fact that the likelihood of a transition cow experiencing an immune insult is far greater in early lactation than it is at any other stage of the lactation cycle, right? There is a lot going on in the time around calving, and even really looking from dry off to that early postpartum period. The mammary gland involution at dry off, that's an inflammatory event. There's a lot of pen moves that are occurring. There's dietary changes. There's the parturition process itself, expulsion of fetal membranes, uterine involution. These are all events that are capable of stimulating immune activation and inflammation. And we know now, after 15 to 20 years of research, that all cows do have inflammation in this time frame, which is not surprising, and some of that inflammation is really important, right? We need inflammation, but it can get out of hand because there are so many events that can contribute to immune activation. And this has been a huge area of interest, as many people likely know. We see that when animals are exposed to a stacking of stressors, so on top of that normal inflammatory, on top of those normal inflammatory events, when they have additional management stressors, this creates this hyperinflammation that has a huge impact on transition cow success, and thus has been a huge area of interest. And I'm just going to highlight a few of those examples here in the next few slides. Erminio Trevisi has done a lot of great work in the area of hyperinflammation in the transition period, and this is the results of one of the studies where they characterized inflammation using a liver activity index. And cows that had a greater degree of inflammation, not surprisingly, produced less milk across the first 4 weeks of lactation. The increase or the reduction in milk yield that occurs in these inflamed cows is likely a reflection of lower dry matter intake, because we know inflammatory mediators have very potent effects on intake, but also coupled with the fact that animals that are in an immune activated state have an increased energy requirement to fuel immune cells that are activated. So this is likely a reflection of both of those things. And with that, Erminio Trevisi's group has also shown that cows that had that greater degree of inflammation also had higher levels of mucus and ketones. Again, because they're consuming less dry matter intake, they have greater energy requirements, and they're still trying to produce milk. So they're in a greater energy deficit as a result of inflammation. Likely this is the case rather than these changes in energy balance actually going on to cause poor transition cow success. They're more likely then to be the reflection. And it's likely the same relationship exists in the space of calcium. Jess McArt's group out of Cornell has done a lot of great work looking at the relationship between, or these different classifications of calcium status. And they've defined this group of dyscalcemic cows that are have a more persistent or even a delayed reduction in calcium status. And recently they published research that shows that those cows with dyscalcemia also have a heightened degree of inflammation. Again, can't say what came first, the chicken or the egg, but we think based off of our findings from LPS studies that this is likely a reflection of inflammation causing disrupted calcium homeostasis as opposed to the reverse. Based off of recent evidence, the Baumgart group, we've recently proposed that we need to rethink the transition period and start to think about inflammation as kind of the starting point that dictates transition cal success. It's now been linked very well to impairments in reproductive performance, changes in calcium status, impacts on milk yield and dry matter intake, and then ultimately can go on to also impact that risk of metabolic and even infectious diseases. In conclusion, cows in early lactation actually mounted a more robust immune response to LPS when compared to mid-lactation cows, which really suggests that they are not immunosuppressed, and immunosuppression is not the reason why our transition cows struggle. They're actually fully capable of mounting an immune response. It's more likely that the reason our transition cows struggle is because they are exposed to so many immune insults that creates this hyperinflammation that then dictates transition cow success. So the million-dollar question of this space is, what exactly do we do about it? How can we help? And I think this is always going to start with management. We know we need inflammation. Inflammation and the immune activation becomes a problem when there's a lot of stressors stacked on top of each other. So we need to do everything in our power to limit insult exposure, control what we can control, utilize strategies to reduce things like mycotoxin challenges, make sure pen moves have a good purpose and we're not moving cows more often than we need to, make sure dietary changes are not too drastic, and make sure we're doing the basics, right? Heat abatement, keeping feed in front of cows, etc. When we can't limit exposure, there's still opportunity to help hasten the resolution of inflammation. And I think this is an exciting area, especially in the dietary feed additive space. I'm very excited to have been here on this webinar with all of you today, and appreciate D&D Ingredients for the opportunity. Would love to connect with anyone on this content if they have any questions. My information will be shared. Thank you very much.
Thanks for tuning in to the Dairy Knowledge Exchange. Don't forget to check our product page for featured products and promotions throughout the year. Connect with us on LinkedIn or share your thoughts and topic suggestions directly with me at pat@ddingredient.com. We look forward to hearing from you.